Hello everyone, I'm Komal. Welcome to InGrade learning platform NEET JEE Foundation, where you learn, practice, crack, succeed, stay focused, stay unstoppable. So students, in last class we have studied about the nucleotides. What we have studied in nucleotides is, wait. Okay, so in nucleotides, what we have studied is we have studied about the components of nucleotides, that was pentose sugar, phosphate group, and nitrogenous bases. We have studied the types of nitrogenous bases. We have studied the structures of nitrogenous bases, and जो purine and pyrimidine था, ठीक है, we have studied their structures. Okay, so So what I've told you in the last class was that nucleotides are basically the monomers of nucleic acids. Okay, so now today we have to discuss the structure of nucleic acids. We have to study about the details of nucleic acids, but before that one topic was left, that was higher nucleotides. So firstly we will study about the higher nucleotides. What are higher nucleotides? See, okay, see students, we have studied that in nucleotides we have three components, right? We have three components. What are the three components? We have sugar, okay, which was attached with the nitrogenous bases, and it was attached with the phosphate group. Okay, if we have only single phosphate group, it becomes monophosphate. Okay, let's say if it is adenine, okay, and it is attached with sugar, and then here is one phosphate which is attached with the sugar, then we'll call it, this nucleotide will be known as adenosine monophosphate. Okay, if we have two phosphate here, it will become adenosine diphosphate. Okay, and if we have three phosphate groups here, it will become adenosine triphosphate. So what are higher nucleotides? These are the nucleotides. What are higher nucleotides? Higher nucleotides is basically nucleotides that have more than one phosphate group, phosphate group, that are known as higher nucleotide. For example, we have adenosine triphosphate. ATP is one of such examples. This is adenosine triphosphate. Adenosine triphosphate. Okay, now if we discuss the structure of adenosine triphosphate, then what we have is, see, we have ribose sugar here, okay, we have adenine group here, okay, we have adenine group here, okay. Now in adenine, the hydrogen which is involved in the glycosidic bonding is the hydrogen which is attached with the 9th nitrogen of adenine, as I've told you in previous class. So it will make the bond with the sugar, it will make the bond with the sugar, and this bond will be glycosidic bond. We have discussed the bondings in detail in the previous class. So if you are not able to understand this, please firstly watch the first class, the previous class, then this. Okay, so you will come to know how this bond has been formed. Okay, now we'll join 3 phosphate groups here. Okay, firstly okay, we'll make 3 phosphate groups here then. Now we'll make the bond here. Let's see how we'll make the bond. Okay. Let's see now. What we'll do is, firstly, wait. Here we have on the 5th group, 5th carbon, we have CH2OH here. CH2OH here, okay, which get hydrolyzed, which get eliminated as water molecule here. And it will form ester bond. This will form ester bond here with this. Okay. Then we have this OH group and H group here. It will form high energy bond with this phosphate group. Okay. And then we have this. It will also form high energy bonds here. Okay, so these phosphate groups are basically joined with each other through high energy bonds, and an ATP, when we call the ATP as energy currency of the cell. Why we call them energy currency of the cell? As the energy gets stored in these high energy bonds, okay, that are basically connecting the 3 phosphate groups here. So this is the structure of ATP. This is the structure of ATP. Okay, class. So that is all for the higher nucleotide. Now we can start with the nucleic acid, that is our main topic. Okay, so let's start with the nucleic acid now. So in nucleic acids, first we will talk about the DNA. Okay, so what are nucleic acids? Nucleic acids are basically polymers of nucleotides. They are polymers of nucleotides. Now if I discuss about DNA, right? So DNA is basically, when it was discovered, DNA was discovered in WBCs, in nuclei of WBCs, in 1869. Okay, it was discovered in nuclei of WBCs in 1869. Now if we discuss about the now if we discuss about the nucleotides, right, how nucleotides are, like all the nucleotides are arranged in nucleic acids. So we will discuss about that first. Okay, so that is the phosphodiester bond, how nucleotides are get arranged in nucleic acids. Okay, so this we have is our, okay, let me draw it for you. For example, we have we have here is sugar, then we have phosphate. This is a, I'm drawing a single strand only. We have sugar, we have phosphate, we have sugar, we have phosphate, then we have any nitrogenous base, say adenine. Okay, then we again have sugar, phosphate, then we have again nitrogenous base, say guanine. Then we have sugar, phosphate, then we have say any other nitrogenous base, say cytosine. Okay, so these are the different nucleotides. Now how these nucleotides are bonded with each other in nucleic acids? They have bonds known as phosphodiester bond. Okay, these bonds are known as, they are known as phosphodiester bonds. These red bonds that I have made is phosphodiester bonds. Now how they are formed, let's see. How phosphodiester bonds are formed, let's see. Let's say we have a sugar here. Then we have a base here. Okay. Then we have a phosphate group here. Okay, so this is the ester bond, right? This bond is ester bond. Now let's make second nucleotide here. We have a base here. Okay, then we have a CH2 here. Then we have a phosphate group here. Okay class, now how these two nucleotides will get attached with each other? So firstly, if we talk about the second nucleotide, okay, so it's one of the hydroxyl group, is making ester bond with the fifth carbon of the second nitrogenous base. Okay, okay, so if I talk about the other bonding that will join these two nucleotides, okay, how it will get attached? This particular phosphate group which is attached with the fifth carbon of the second sugar, okay, it will also form the ester bond with the previous sugar, with the third carbon of the previous sugar. Okay, for example, we have this nucleotide number 1 here and we have HOH here, right? Okay, it will get formed here. So this particular water of molecule will get eliminated here, and here it will form ester bond. Here it will form ester bond with this. Okay, now this particular phosphorus have ester bond with the fifth carbon of the next sugar. Okay, and this particular phosphate have also ester bond with the third carbon of previous sugar also. So this particular phosphate group have two ester bonds. Okay, one with its own sugar, one with its fifth carbon of its own sugar, and the third carbon of the previous sugar. Okay, so this is also ester bond, and this is also ester bond. So this whole bond is known as phosphoester bond. This bonding is known as phosphodiester bond. Phosphodiester bond. Okay, class? So this is how we make phosphodiester bond. Okay? Chalo. So this is what I've told you, that it is the phosphodiester bond. Okay, let's discuss further, that is Watson and Crick model. What is Watson and Crick model? Let's discuss Watson and Crick model. Basically, it's a model of the, it describes the structure of bDNA. This model describes the structure of bDNA. Firstly note this down, class. Note this down. Okay, class. Next, we'll discuss about the structure of bDNA that was explained by the Watson and Crick model. Okay, let's see. So Watson and Crick model was given in 1953. Okay, it was given in 1953 by Watson and Crick. By Watson and Crick. Okay, now what this tells us about the DNA model? Firstly, it tells us about DNA, that is that it have a DNA have a helical structure. DNA is double-stranded. Helical structure. DNA is double-stranded helical structure. Okay, that means it is a secondary structure, as we have discussed in proteins. Secondary structure, that is alpha helix structure. Okay, both the strands are anti-parallel in direction. Both the strands are anti-parallel in direction. What is the meaning of anti-parallel in direction? That means the 5' end of one strand faces the 3' end of complementary strand. What does that mean now? Let's discuss this. Let's discuss this point first. And also it tells us that both the strands are unbranched. Both the strands are unbranched. Okay, both the strands are unbranched, right? Okay, class. Now let's discuss this point, this 5 dash and 3 dash point. You must not be able to understand this without a diagram. So what is the 5 dash and 3 dash end in the strand? See, I have told you that we have all the nucleotides attached, or they are aligned in a nucleic acid with the help of phosphodiester bonds, right? Let's discuss this. We have one strand and we have a double stranded DNA. Okay, so let's make it. We have sugar, we have any base, say adenine, we have phosphate. Okay, then we have again any sugar, this is second nucleotide, this is phosphate, this is guanine, then again we have sugar, phosphate, cytosine. Okay, now first of all they are anti-parallel in direction. If this is 5 dash to 3 dash and 5 dash to 3 dash end, okay, then this strand will be anti-parallel in direction. For example, we have cytosine, in front of cytosine we'll always have guanine. Okay, so we have here sugar and this is phosphate. Okay, and we have triple bonds in between them. We have triple bonds in between them. Then again we have complementary sugar. If we have guanine, we have cytosine always in front of this, and we have triple bonds in between them. Okay, we have sugar, we have phosphate here. Okay, then we have again in front of adenine we have always thymine, and we have double bond in between them attached with the sugar and phosphate. Okay, so these are the complementary nucleotides in front of each other, facing each other. Okay, they have phosphodiester bonds between them. Okay, now what decides that which end will be the 5 dash end and which end will be the 3 dash end? So how it is decided? So basically, if the 5 dash end of the sugar, say if we have this 5 dash end of the sugar, let's say this is the first nucleotide. Okay, if the 5 dash end F of the sugar is free, that is not attached with any phosphate, okay, not attached with any phosphate, this will become your 5 dash end. Okay, this will become your 5 dash end. And if as we go down, the last nucleotide, in last nucleotide the 3 dash carbon, third carbon is free, that is which is not attached with any phosphate, so it will become 3 dash end. And if the chain will become anti-parallel, this will become 5 dash end. This will become 5 dash end, and this becomes the 3 dash end. Okay, so that's why it is said that 5 dash end of one strand faces the 3 dash end of the complementary strand. Okay, class? Okay, let's discuss further now. Second point which is told us is that the strands are not coiled upon each other. As we have seen in many pictures or in many diagrams, the strand of DNA looks something like this. So it makes an illusion, it creates an illusion like they are coiled upon each other. Okay, so Watson and Crick model explained this as that both the strands do not coil each other, are not coiled upon each other, instead they are coiled around a common axis, like a helical staircase. You must have seen a helical staircase in many homes like this. Okay, and so basically there is this common axis around which both the strands are coiled, in which one strand have 5' to 3' end and other one has 3' to 5' end. Okay class, now it has compared the structure of DNA with the helical staircase, okay, in which the backbone, this all is the backbone, this is the backbone of DNA. Okay, so it is compared to the railings of staircase, helical staircase, in which backbone of DNA is railings of staircase, okay, and the steps and the nitrogenous bases are compared to the steps of the staircase. Nitrogenous bases are steps of staircase. I hope I have made you very much clear with this point. This is the backbone of DNA. Okay class. Okay, so this was the whole second point. Okay, now let's discuss further points. Next point, which is explained by the Watson and Crick, was the backbone of DNA. The backbone of DNA is made up of alternative sugar and phosphate components, as we have already seen. Then again I have told you the next point, that the strands are complementary. So both the strands are complementary. What does that mean? That means cytosine always faces adenine. Oh sorry, thymine always faces adenine. cytosine always faces guanine. Okay, and vice versa. Okay, both the strands are stabilized by hydrogen bonds. Both the strands are stabilized by hydrogen bonds, as we have already seen, stabilized by hydrogen bonds, in which thymine and adenine have 2 hydrogen bonds and guanine and cytosine have 3 hydrogen bonds. Okay, they have 2 hydrogen bonds here. They have 2 hydrogen bonds and they have 3 hydrogen bonds. Okay, class? Next point which is explained by the Watson and Crick is that the nitrogen bases are nearly perpendicular to the backbone of DNA. Okay, for example, if we have sugar, we have phosphate here, the nitrogen bases are always at the 90-degree angle. Okay, next up we have some data. We have the basic data of the DNA. What they say is, they say that suppose we have a, let's say we have a we have the DNA. Okay, this is one helical turn. Like this. We have one helical turn. This is one helical turn. Okay, then they say one helical turn have one helical turn have 10 base pairs in bDNA. In bDNA, we have total 10 base pairs. Okay, let's make 10 base pairs. We have adenine, we have thymine, they have double bond. We have guanine, cytosine, triple bonds. We have again thymine, adenine, double bond. Then we have cytosine, guanine, triple bonds. Then we have thymine, adenine, double bonds. Again we have adenine, thymine, double bonds. 2, 3, 4, 5, 6, 7. Guanine, cytosine, triple bonds. Cytosine, guanine, triple bond. Adenine, thymine, double bond. Okay, so like this we have 10 base pairs, okay, in one helical turn. Okay, now they say that interbase pair, like the distance between two base pairs, this is two base pairs, these are two base pairs, and the distance between two base pairs, interbase distance interbase distance. We have interbase distance equals to 3.4 angstrom, or you can say it is 0.34 nm. Okay. So if we have 10 base pairs, pitch of DNA or length of DNA, how we can calculate the pitch of DNA or length of DNA? If we have interbase pair distance is 3.4 angstrom, then we'll multiply it with the 10. 3.4 angstrom, it will become 34 angstrom, or it will become 3.4 nm. So it will be the whole length of the DNA. Okay. Then we have is the distance between two strands is 20 angstrom or 2 nm. That is diameter of the DNA. We have diameter of DNA, that is 20 angstrom or 2 nm. Then it also explains the angle between the base pairs. The angle between two base pairs, say this is the angle between two base pairs, is 36 degree. It is 36 degree. How it is 36 degree? Because turn of one helix is equals to 360 degree, and we have 10 base pairs, so we'll divide it with, it will divide by 10, so it will come up with the 36 degrees. Okay class, so this was the whole Watson and Crick model DNA. These are the all points that were explained by the Watson and Crick. Okay. Now let's discuss the Chargaff's rule. What is Chargaff's rule? These were the rule given by Erwin Chargaff. In 1950. Okay. What he told is that amount of purine is always equals to amount of pyrimidines. Amount of purines that is A plus G is equal to amount of pyrimidines. That is T plus C. Okay. Then the next point he told us that amount of adenine equals to amount of thymine. And amount of guanine equals to amount of cytosine. Okay class. Third point he explained us that this base ratio is A plus T upon G plus C. Okay. This is specific for it remains constant for specific, for same species but varies for different species. Okay, that it is constant for if it is more than one, say it is more than one for humans, it will remain more than one for every human, okay, with every homo sapien, okay, but it will, if we compare it between the homo sapien or lion, it will be different, okay, this base ratio will be different. Also it tells us that if a plus t, if this ratio a plus t upon g plus c ratio is less than 1, is less than 1, okay, that means g plus c, amount of g plus c is more than a plus t, which means it have, which means structure have more H bonds, and more the H bond is, more is the stability. So structure will be more stable. Okay, class? Fourth point which is explained by Chargaff rule was that sugar and phosphate moieties are in equal proportion. Okay. Now Chargaff rule is only applicable to DNA and it is not applicable on RNA. This is very important point which is explained by NCERT, that Chargaff rule is not applicable on RNA. Because, what is the reason? Because most of the RNA is single stranded, because of its single stranded nature. And also we have uracil in RNA instead of thymine, so it has lack of complementarity and lack of complementarity. That's why Chargaff rule is not applicable on RNA. Okay, class. So I hope I have made myself clear with the Chargaff rule now. Let's discuss about RNA. RNA is ribonucleic acids. Ribonucleic acid, mostly present in RNA. Mostly present in viruses. It is a genetic material of virus. Okay. Mostly RNA are single stranded. Most of the RNA are single stranded. Exceptions that we have is reovirus and dwarf rice virus, as they are double stranded. They both are double stranded. They have double stranded RNA. Okay. Okay, class. Now let's discuss about the types of RNA. Types of RNA. We have three types of RNA. That is, we have messenger RNA or mRNA. Messenger RNA or mRNA. Acts as template protein synthesis. Next up we have is ribosomal RNA. Or rRNA. They are largest type of RNA. Largest RNA constitutes nearly about 80% of total RNA. Also have role in protein synthesis. Next we have is tRNA or transfer RNA. Okay, tRNA is smallest RNA, constitutes nearly about 10 to 15% of total RNA, and they transfers amino acids for protein synthesis. Okay class, so this is the types of RNA. Next we have is types of DNA. Types of DNA, we have 5 types of DNA. We have 5 types of DNA. That is